I'm Shannon Hughes. I'm a program director in the Division of Cancer Biology, and today I'm here on behalf of the Generation of Human Tumor Atlases implementation team. Over the course of the last couple decades, we've all come to realize that there's overwhelming complexity within the tumor ecosystem. And thanks to initiatives such as the Cancer Genome Atlas and related projects, we have a better understanding of what the genetic underpinnings of cancer are. However, how the cellular and non-cellular components within this tumor ecosystem interact to drive tumorigenesis is less well known. And our gaps in understanding of this make it difficult to predict prognosis or to develop risk strategies and also strategies for precision screening and new treatments. If we were to have a much more comprehensive view of the tumor ecosystem, including a better understanding of the cell types involved and their proximity to one another in terms of how they interact, this could facilitate a better understanding of tumor heterogeneity and tumor evolution that gives rise to such tumor heterogeneity. Additionally, understanding the contribution of non-tumor components, such as the stroma and, importantly, the immune cells, including the extracellular matrix, we would be able to identify better markers of progression and drug resistance, and also possibly to develop early intervention strategies and robust therapies. New and transformative technologies and computational approaches can now facilitate such a comprehensive view of cancer. Bulk analysis has and will continue to provide a significant amount of important information about tumorigenesis. However, new technologies that are more at the single cell or near single cell level, such as single cell transcriptomics, allow us to have a much broader view of the components within the tumor environment, such as this paper described in 2016 regarding the different components of metastatic melanoma. You can also look at this from the protein level with new technologies such as mass cytometry, in which now millions of immune cells have been profiled across a couple of different tumors. Two recent papers in clear cell renal cell carcinoma and early stage of lung cancer have shown that there are a variety of new immune populations that we didn't otherwise appreciate within the tumor. And these types of populations transition over the course of a tumor lifetime. And both of these papers pointed out that these new populations might be predictive of patient outcome, although more research is needed. Importantly, the two examples I just gave you underscore dissociated types of analysis, but we really need to also understand how these cells are in relation to one another within the tumor microenvironment to really understand how they interact. 3 working groups of the Blue Ribbon Panel appreciated the need for such data and recommended the generation of human tumor atlases. With regards to these panels, their definition of a human tumor atlas was really a high-resolution map that showed the dynamic, so with time, 3D architecture of an individual tumor. It provides molecular, cellular, and physiological information, not only about the individual cancer cells, the cancer mass itself, but also the tissue of origin and sites of metastasis. Importantly, information can be gained about how the tumor affects the immune system and vice versa. The main goal of building a human tumor atlas is to enable predictive modeling that can help to refine therapeutic choices and other choices for patients. And within these working group recommendations, 3 different specific time points were suggested, including the transition from pre-malignancy to full-blown cancer, from invasive to metastatic disease, and in response to and during development of resistance to therapy. The working groups recommended pilot atlases in exemplary pediatric and adult cancers, including at least one atlas that describes a cancer that's responsive to immunotherapy and one that describes a cancer where the responses have been poor. In response to these recommendations, we proposed the Human Tumor Atlas Network, or the HTA Network. And the goal of the HTA Network is to generate pilot-scale, high-priority human tumor atlases that facilitate basic and clinical scientific discovery regarding important transitions during tumorigenesis, and these are the 3 transitions that I just explained to you. We propose 4 components of this network, including 2 different types of research centers, the HTA Research Centers, which will focus on the later transitions in cancer, and the Pre-Cancer Atlas Research Centers that will focus on pre-malignancy. These research centers will be coordinated by a set of coordinating centers, which are incredibly important for bridging the different research centers, as was brought up during the discussion of the last RFA. Importantly, a variety of high-priority cancers have been identified as the first targets for this pilot-scale effort. And these high-priority tumor types include tumors that are responsive or non-responsive to immunotherapy, those that are highly metastatic or highly refractory to therapy. They also include tumors that are representative of cancer disparities, and tumors that are driven by genetic aberrations such as BRCA1/2 mutation or Lynch syndrome in colon cancer. Just very recently, there is a pre-cancer atlas think tank in which the participants of that think tank, some of which are in the room today, really prioritize not the tumors themselves, but the types of ideas behind the choice of tumors, and those are really represented in this list. Thank you to Dr. Tubison, Parada, and Willman for suggesting that we give a little bit more information about the makeup of the research centers themselves. We envision the research centers to be made up of multi-investigators and interdisciplinary investigators that can span the entire range of tumor atlas building activities. From those who are experts in biospecimen acquisition, now specifically biospecimens that will address the tumor atlas that's being built within that center, so it's addressing a specific transition in cancer. And within this biospecimen acquisition, processing, and classification unit, there will be pathologists, clinical oncologists, and other experts in that field. Those biospecimens will be passed to the molecular, cellular, and tissue characterization unit for multi-parameter, multi-scale analysis. This could be at the single cell level or above. We're not being prescriptive about the type of tumor cell atlas that's going to be best representative in each one of these transitions. The data, which will there be significant data, will be passed to the data processing, analysis, and modeling unit. And this unit will have the responsibility for not only analyzing the data, but constructing a final tumor atlas from each center. As I stated before, the network will be coordinated by two coordinating centers, a data coordinating center, which we funded or we request funding for in FY18, and a tissue coordinating center, which we request funding for, we will request funding for in FY19. I want to give you a little bit of information about the responsibilities of these coordinating centers because they are very important, not only for mediating interaction between the research centers, but also across the Cancer Moonshot efforts and those outside of the Moonshot. So the Data Coordinating Center will be responsible for coordinating policy and science-focused working groups, so for example, common data elements, et cetera. They'll provide some administrative support, but most importantly, they'll coordinate the data flow within the network, and I'm going to talk about that on the next slide. The Tissue Coordinating Center, which we envision to be a virtual center funded in FY19 based on the types of tissues that will be collected based on the atlases that are proposed, will oversee tracking and distribution of these biospecimens, not only within our network, but outside of the network. This is the proposed data flow for the HTA network. So we recognize that a significant amount of data is about to be generated by these research centers. And we expect the data to flow into the HTA Data Coordinating Center to be immediately available back to the different research centers. The Data Coordinating Center will also facilitate data deposition in publicly available data commons, for example, the NCI Genomic Data Commons or the Cancer Imaging Archive. And we've partnered with another Moonshot implementation team in order to propose a cancer data aggregator, which will facilitate the integration of what's likely to be very disparate types of data based on the types of atlases that are built. The data will be also available to the community and shuttled back into the research centers for final construction of atlases. We recognize that right now we may not have all of the technologies available to us to build the most optimal atlases, so therefore we'll be partnering with the Enabling Technologies Cancer Moonshot implementation team who will hold workshops with our investigators to determine where holes lie, and then using administrative supplements to existing NIH grants bring in new technologies that can broaden the atlas. After a pilot of 5 years, we envision 5 live tumor atlases that are intuitive, searchable, and informative for both the research, clinical, and possibly the patient communities. These atlases will be built upon a searchable database of harmonized, multi-scale data that's easily accessible to the research community. In addition to the atlases, there will also be a validated set of SOPs for tissue acquisition, data collection, and open source workflows for analysis. With these preliminary set of comprehensive human tumor atlases, the types of questions that we can answer are now, can we really define what we mean by tumor heterogeneity within and across the patient population? And can we quantify the dynamics and the 3D architecture of the tumor ecosystem? And most importantly, facilitate predictive modeling that leads to better patient outcomes. Looking across the NIH portfolio, there are really no grants that specifically address an entire tumor atlas, although there are many programs and individual grants that can provide certain aspects of atlas building activities. And we hope that to partner with these different programs across NCI, along with programs that are being supported by NIH, such as the HubMap Common Fund program, which will be building atlases of normal tissue, and programs that are outside of NIH, such as the Human Cell Atlas Consortium, and also the more recently funded programs from Cancer Research UK. We request 5 research centers with a level of funding at $2.35 million in direct costs per year, and also funding for our data coordinating center, which will increase over the course of the lifetime in order to accommodate all the data that's being generated by the network. With that, I'd like to thank you and our BSA reviewers, along with the whole generation of human tumor atlases team, which is very trans NIH. And finally, I'd take any questions that you have. Thank you.
Okay, well, thank you, Dave. Okay, thank you, Chi and Shannon. So as stated, the Human Tumor Atlas project is meant to be an intuitive, searchable, informative, dynamic, and moist deliverable. And by that I mean there actually is tissue generated in the end, which will be shared amongst the groups that are participating, and as written, may be available for the community later. So it's not just a static entity. So it's like going to Epcot Center and getting that spray of water on you at the same time. But Epcot is a good analogy. You're going to be put in this place and see what it's like to be inside a tumor and all the cell types and as they dynamically interact, et cetera. And so when we had the first phone call with the group, I made the point that it didn't seem like the call was written by pathologists. It was written by biologists. And what pathology has taught us is that if you look at autopsies for people who didn't die of cancer, you find pre-neoplasms. You find lots of pre-invasive cancers in certain tissues. And that immediately tells you that there are lots of pre-invasive neoplasms which actually will not progress. The vast majority won't in many of the tissues, including some of the ones which are highlighted here. And so we had a conversation about would it be possible that rapid autopsies could be included in this call, because then we could actually see the A to Z on many of these diseases, look at patients who die of a particular cancer and actually be able to reconstruct possibly the malignancy by finding pre-invasive neoplasms in those patients and have access actually to all the tissues that are going to be necessary to actually do this type of work. So to get enough tissue sample to really get granular information, you need access to big pieces of tissue, not needle biopsies. You would need blood, the tumor, obviously, the surrounding tissue, maybe the other tissues of their body if you really want to understand this process. Shannon and her colleagues were receptive to this concept, and in the longer write-up you'll see that autopsy specimens would be thought of as potential candidates. So again, the notion though is to create a comprehensive atlas using the latest technologies, and these would be prospective samples because the latest technologies have not been used on TCGA, have not been used on most of our other data sets. They'll be informative, but they won't replace what we're trying to do here. And the deliverables are to understand, I guess, the steps involved in cancer initiation, progression, metastasis, but also to interrogate the microenvironment. And for the immunologists here, we just had an immunology presentation, that's what they always ask for, is much better sample sets so that they can see what's happening. So there are new technologies, single-cell analysis. There are great examples. I'm sitting next to someone who has a cool single-cell analysis on his laptop right now, Tim Lay. And I'm doing it, and it bankrupts me. It is really expensive. And I think Liz was making this point about T-cell receptor sequencing. These new technologies are way more expensive than the other ones we're talking about. So, again, eyes wide open. We have to be aware of that. So physique is a new method to do in situ probing of tissue. It's sort of a different way of multiplexing antibodies, but you can actually do sequencing of single cells at the transcriptome level. It's very low throughput, but very high content. And one thing, again, not mentioned here, but if we're going to have dynamic models available, that necessitates having timed biopsies or something you're reconstituting from a patient. So there will have to be models created, not mouse models that Kevin and I and Tim make, but rather tissue models where we reconstitute the immune cells, the stromal cells with the neoplastic cells, and then use them to ask questions. And I guess we're leaving it up to the creativity of the applicants to talk about that, but I do think that could be added maybe to the language, that models should be a deliverable from this, human models. The way that this is constructed, I think will allow pathologists to maybe come back to the table. They're the ones who created our field a long time ago before we could clone genes, and they think a lot about these issues, and I would love to see them be major participants in this call, along with surgeons and oncologists, working with biologists and computational scientists, as mentioned. The pre-invasive cancer meeting, I couldn't attend it, but Liz attended it, and I don't know how many other folks here were able to attend it. Sounded like it was really interesting. I don't know why pancreas cancer is not on your list. To me, it has the most advanced tumor
I tried to make the case, but...
It's the most advanced tumor microenvironment of any cancer.
metastasizes at the pre-malignant stage quite often.
I don't know why it got the short straw, but you might want to rethink that. It's a missed opportunity. I am conflicted in that. But I guess, again, the last thing is, in the end, you want an encyclopedia. You want to know, how did cancer start, where is it going, and how can we intercept this, either to prevent it, treat it early, treat it later. And I think, again, to do this properly, it will take a real deep dive on some cancers with enough samples to get get proper information back. And of course, technologies will continue to develop, but my gestalt is 10 autopsies is going to teach us more than 100 biopsies, a lot more. And I guess it's a term that we haven't used in this meeting before, but that's what many people have been asking for a long time. I've never seen support for it other than at Hopkins or at Nebraska and maybe a few other places, Memorial now. And I think that would really add a lot to this arena. But in any case, 3 groups wanted it, and I think it will be really informative for the field. But let's not study the pre-invasive cancers that will not progress, because we will be wasting our patients and our taxpayers' time and money.
Luis, yes, I have things to ask.
Well, that's a hard act to follow. Dave, I prefer biologists to pathologists, even though I'm sure pathology is a terrific discipline. I don't think they discovered cancer, they just described it. They just described it, and often they described it wrongly. So I don't think that...
Them's fights words.
I don't think we should move away from the biology that much. And I do believe that it is very complicated, and it is very expensive and that this is new technology. These are all real issues. But the fact of the matter is that it's being done as we speak in real time by people like Aviv Regev and by others, Mario Suva, et cetera. It's happening, as you say, in your lab and in my lab. And I think that if we can bring together dream teams, we can do this in the most logical way. And rather than thinking about all the things that are stumbling blocks, I think it's an opportunity. And I think that since it's being done, if we put the best people together and permit them to do these things, that will work great. I've tried working with autopsies. Most of my cells are dead and I get crap. So, I mean, I think there's a lot of technology to be figured out in autopsy studies before we can really say that they're as valuable as biopsies. But I certainly think we can learn a lot from biopsies. And I think that if we have biologists and pathologists and molecular biologists and computer biologists and all of them together, that would make the best of this possible world. But what I think what is mostly important about this approach right now is that if it's not embraced by the community and by the NCI, that we will have all these outlying pockets that will present their data in an even more biased way than what could happen. And so I think it's a great opportunity to incorporate and to bring together the best people to try to do this as well as possible, given what we know and what we have.
So before I open it up for discussion, Dave, you want to make a motion on this?
Yeah. So again, my comments were meant to stimulate thinking before we just, we're towards the end of the day and she can push us around easily. I'm supportive actually of this motion. I'm just trying to improve it.
But you can make that motion to include what you want.
Well, they already included the wording about autopsies, but I wanted Luis to actually say something, so he did. So I motion to support this proposal.
Okay. Second. Yeah. Okay. Any further discussion?
Yeah, there's a computational pathobiologist in the audience here. So, I just want to put a point on it. I think that TCGA has done an amazing thing for cancer research, and this is the evolution of the TCGA program. So I do share skepticism about the use of autopsy tissue because of the well-known lysis of inflammatory cells that happen in autopsy samples. Tumors intact, stroma's intact, but inflammatory cells are significantly harmed, so I'd be cautious about using autopsy materials, and I am an anatomic pathologist a long time ago. The other part about this is that, if you want to have the impact of TCGA, unfortunately we don't have the money TCGA had, this is significantly underpowered. And if there's ways to grow and expand this program down the road, it will pay really big dividends. So kudos on this.
I just wanted to comment about the informatics part. In order to get maximum, I think, bang for the buck here, and since it's focused on the human tissues, it's going to be really important to have good connections to the mouse model systems informatically. I don't mean that they should be supported by this mechanism, but to the extent that we really want to understand the biology of the interaction of these cells and that there are really good mouse models that have been correct for a number of diseases, at least to compare, and the same technologies are being applied of single cells and of the immune system and interactions within these animal models. I'd like to see a mechanism, and whether it's within the mouse models consortium, if that still exists, or a mechanism to be able to connect to the mouse models informatically to pull out the data and compare them, because if you find that the human and the mouse have these similarities, the mouse is tractable to experimental manipulation, and then you could look at function as well as just categorizing an atlas. The atlas is good, I think, as the start, but what we really want is the function of these cells and how they interact.
Two errors in the TCGA that should not be repeated here. One is exquisite detail to clinical annotation for all patients who are studied. And the second is serial sampling. And attention to those two things will make this so much more powerful and this has to be decided upon up front, and adequate and considerable resources are needed to do both of those things.
Yeah, two comments that follow up on the informatics discussion, the TCGA. Could you go back to your proposed data flow slide,
Sure.
please? And you've got a schematic in there where you're sort of pushing data from the aggregator out to the community. Is that how you envision this to happen? Sort of a data distribution component?
I think so. So the aggregator is very much still a work in progress, but the way what we're envisioning it is that it would be something that the community could access or build upon themselves to create gateways into the aggregator to analyze the data in the way they want to.
Yeah. I think that raises a real question of whether this can scale up at the end of 5 years. And the other reason that I say that is your budget has increasing data costs in the out years. And in fact, we should be looking for just the opposite. Building an infrastructure up front that is inherently scalable and that brings people in as you add in the other components, to me is the way to go. It's more consistent with the data commons. So I'd encourage you as you develop the RFA to particularly talk to the informatics folks, the cancer data commons folks, Warren's group, and really see if you can shift that model for this to be a scalable, bring the community in, provide the resources, and I think that's the way you're going to get better progress 5 years in and then scalable long term.
So I agree with you at one level, Jim, but I think the other side is we do want all the data to flow into the data commons. Where this funding really is focused is how do we define the standards and how do we actually help the teams that are generating the data do the curation, annotation of it. And that's something, as we start generating more data, is unfortunately has to scale with the data generation. So although the where it's going to be stored is, I think you're right, it needs to be highly scalable and relatively expensive or inexpensive. The cost of bringing that data in and defining the standards around it and doing that work of annotating it, that scales with the data generators.
Cheryl?
So I want to understand the prescriptive nature around the high priority tumors. So anything that's not on that list would not be responsive, is that correct?
So we thought very carefully about the list that we were putting up today, and also with the understanding that we view this as a pilot where we will be generating pilot atlases within these tumors that not only inform us about the tumor types or the questions that are being asked within those tumors, but also what the best type of atlas would be to move forward. So in the next stage of a program, you would then focus on a certain type of atlas that was most informative potentially, and then scale to a greater number of tumors. So
you might want to reconsider that. I think you heard a case about pancreatic. You know, the Chevier cell paper you cited there was on renal cell carcinoma, which is another example. I mean, the other way would, of course, let your applicants make the best case argument about why their particular tumor would be a good one to use rather than a priori saying up front it's this or nothing.
Yeah, I totally agree with you, and I think that that's right. One of the constraints that we had was thinking about how we really want to have some great crosstalk across these research centers. So by limiting the total number of tumors that we put in this RFA, we might be able to have a better collaboration in this initial pilot.
So you could still do that without being prescriptive. You're going to limit the number of tumor applications that you fund.
Charles?
I mentioned this to Shannon before, but I want to make people aware that there's a very synergistic potential effort run by Chan Zuckerberg on a normal cell atlas. They have a lot of resources and money. They've already had 2 big organizational meetings, and they're already starting to fund some projects. I know Shannon's been in touch with them, but I think these groups could come together better. It seems like a lot of us are doing single-cell RNA-seq. There's no doubt in my mind that's going to be the technology for this. And in terms of price, it's absurd. And it's absurd because of one vendor, 10x, that makes this ridiculously expensive kit. And so if there's some way we could do something about that, that would be helpful.
Yes. Yeah, so I forgot to mention one other point that came up in the discussions before this meeting, and that was that there should be measurables with teeth. In other words, there can be great proposals and, we're going to sequence this and we're going to CyTOF that and we're going to do this and we're going to do that. there has to be progress. And one could imagine groups that have great ideas and that just don't coalesce and don't make progress. And so there should be a very strong sort of measurable progress, at which, if there's no progress, funding gets stopped because of the nature of this being a new initiative with new technology and very expensive. One shouldn't run through the many years if a group really isn't. There's always excuses, but at the end of the day, I remember back in the old human genome sequencing days, if you're going to sequence chromosome 16, sequence chromosome 16. You're given a lot of money. We know it's hard and whatever, but other people are getting 15 done and 2 done, so do 16. So there should be teeth on progress reports, and I actually feel very strongly about that.
Vicky, first.
Oh.
I just wanted to stress the value of carefully annotating the data attached to the specimens. We heard this morning, for example, in the PDX model system, that a large percentage of the samples, they didn't even know the ethnicity of the patient donors. So I would strongly recommend a core set of just simple data besides just the clinical and follow-up data on each patient, because this enormously enhances the value of the specimens.
Yeah, Vicky.
So I think the third sin of the TCGA is that it wasn't representative of the population of the United States and didn't include a lot of diversity. So I would hope as you go forward with this potentially that there are metrics that are imposed on the investigators to ensure that you get a wide sampling of the diversity of this country.
Thank you.
Just one comment hearing the discussion. I'm a little unclear about what people think this is. So Shannon said on the one hand, we're going to build an atlas. And to me, what that means is we have the technologies to build the atlas. And the other, she said, well, it's a pilot study. And if it's going to be a pilot study, then I think we should probably go for the lowest hanging, easiest tumor tissues to analyze at the single cell level, which are likely going to be leukemias and lymphomas. If we really think the technology is mature and ready to go, then I think you can look at these more complex tumors. But just listening to the conversation, I'm trying to decide where we stand here. Is this ready for prime time? We just need to make an atlas, or is this we need to do a lot of technology development, in which case we shouldn't be so proscriptive about what tumors we're going to study and not study, because some of the easier ones to study may tell us how to make the technology better before we study the harder ones. And again, this is a lesson from the TCGA with allelic, when you had mixed tissues from solid tumors, a lot of computational algorithms had to be built to figure out what the real allele burden was and what the real tumor burden was, which was much less complicated in the early stages with the hematologic cancers.
Daphne?
So actually very much in line with what Kevin said. I think that we're doing actually an experiment that is called to build the atlas. And if we agree that this is the case, then milestones have to be, the investment is significant. And I just don't think that the deliverables that have been put together here are going to allow us to figure out halfway through, three-quarter way through, whether it's possible. Because this is an experiment that has never been done. We don't know whether the complexity that is going to be generated is going to lend itself to put something together that we're going to be able to leaf through and just say, oh, for this tumor, it's this combination of cells. If you find it in the tumor, that's how the patient will respond to therapy X, Y, or Z. So I will suggest to put something in the RFA that will allow us to dial up or down depending on the progress that has been made.
So I think there's been a lot of confusion, and it was great to have the pre-cancer think tank a couple of days ago, because I think there are people coming from different points of view on having read the Blue Ribbon Panel recommendations, and then finally we came together on what we thought was really the meaning of the Blue Ribbon Panel, which is what I thought was the meaning, and that was really to take the success of the TCGA as well as the challenges and learn from that, and now build on that and include pre-malignancies, include other human cancers, include the tumor microenvironment. I love the way David said it. It is like going to Epcot, and like seeing, looking around and you have the three-dimensional tumor with everything in it. So it's the deliverables are not to say to a patient, okay, we now know how your monocyte interacts with your, gene expressed by this pre-malignant cell. It's more to have for the community data sets that they can now generate hypotheses and move science to the next level. So that would, to me, and so the pilot is to see if we can do that. And we struggled, low-hanging fruit versus, do we take something that's going to be more impactful? And I don't know what the best answer is. And certainly with pre-malignancies, that becomes even more of a challenge because you have even smaller tissue.
So let me just, going back to the question of what it is. So the idea here is basically a repository for single-cell sequencing from many different tumors. I mean, is that the idea?
No, I don't think that's the.
That's just one part of what single cell. I mean, there are lots of technologies that we wanted to include in this.
But it is a repository. People that are going to be doing.
Well, it's more of developing. So very important to this are the pathologists, the computational biologists, everything that people just said, because we want to build a basically three-dimensional picture of different signaling pathways among the tumor microenvironment that includes normal, malignant, inflammation.
So one last comment, and then I'm going to summarize it.
So a lot of discussion has been had about the clinical annotation of the data, and everybody nods, but I find very disturbing when we have a large data set and we're talking about minorities and we don't know the race. And for nowhere could clinical annotation be more important than an atlas. But if we allow this, and it's not in the RFA, that we do something up front to have a minimum data set of collection for clinical annotation, when we come to the end, I hope that we never sit in another meeting like this and they say we have all this data and we don't know race. Like, can't we up front say that there's a minimum part of the call of this, you must have this clinical annotation to go with it, so we'd never do that again.
And that's what we said at the pre-cancer think tank, and that's why some of the cancers were picked. So, and I think either you said it or it was said in the immunotherapy one, but in picking breast cancer, in picking prostate cancer, the biology of those cancers in African Americans versus Caucasians is different. And exactly right, we need to know who they are, and we need to be somehow determine ahead of time what percentage we want. You're absolutely correct.
Okay, so this has been a very healthy discussion, so I'm going to summarize. I think from the subcommittee, there's enthusiasm to the general concept, but as we discuss it, there's a little bit of haziness as to what this really is. For example, should there be a architecture of the atlas being better defined so we know how to populate it? We hear a little bit about whether this is single-cell sequencing effort or what else is going to go into this and how is it going to evolve, right? So do we draw out the architecture ahead of time and then fill it in? Or do we evolve some simple architecture with time to evolve the atlas? Because we don't know what it is. I mean, that's what I'm hearing from the group.
So I think that maybe some of these things weren't described well enough by myself or Shannon. So the notion is that there's going to be an integrative view of a living tumor by recreating it from its parts, either in silico or in your laboratory. But to do so, we need to have access to terrific samples to start with, and there will be clinical information gathered on the patients. I don't know specifically that the ethnicity was mentioned, but it could certainly be included. That's not beyond reach. The technologies being used will be today's technologies with an eye on tomorrow's. So yes, single cell analysis is important because that's how you identify the cell types and the heterogeneity of the cell types. But I suspect that new technologies will replace them that are less expensive, hopefully. And there will be an analysis of the matrix, the acellular matrix, period. I do think that warm autopsies actually are very informative. Maybe Lewis doesn't have a good collaborator for that. And that will add. But the pre-invasive neoplasms, the nice conference that happened last week for the first time apparently ever in the history of the NCI, will be another approach. The only pre-invasive neoplasms that are big enough to really study are going to be DCIS and colon polyps, essentially, and from the solid tumor side. But, so this is a start. It actually is written in here that a pilot has to be established for full funding to go forward. That's written in here, that there will be a pilot project, that shows, form. And a deliverable will be 5 living atlases that will be both, again, in silico as well as in laboratory. And that's their goal. Whether or not more thought, discussion, refinement should happen at this stage prior to considering funding at the Tesla level, I think needs to be determined. But otherwise, I hope that there's not too much ambiguity about what's being proposed.
Okay, and also here that the organ-specific should be relaxed was the other thing. Yeah. Okay, so you heard the discussions. I'm going to have to call for a vote for all those in favor of this RFA. Anyone against? Abstention? Okay.